Okay, everybody, so this is a discussion on acid secretion in the stomach, how it's formed, what kind of things stimulate it, what kind of things inhibited it. Well, acid secretion starts with CO2 and H2O, making H2CO3. As you all know, CO2 can enter all cells, and so they enter the parietal cells of the stomach that secrete the acid. The CO2 will join with H2O with carbonic anhydrase as the catalyst to make carbonic acid. The carbonic acid will then dissociate into hydrogen ion, which will eventually be secreted into the lumen of the stomach, and bicarbonate, which will leave the stomach in exchange for chloride, which will enter the parietal cell using an antiport system. The chloride is allowed to leave the cell from the luminal side using a chloride anionic channel. Parietal cells, which are also known as oxyntic cells or delomorphous cells, in the human stomach are located in the fundus area as well as in the body of the stomach, in the gastric mucosa. They are connected by a lattice of canaliculi, which then allow a constant secretion of acid, which is further triggered by the actions of different regions of the body. We say that the stimulation of the acid beyond the normal sort of baseline secretion is said to be from three phases: cephalic, gastric, and enteric phases. Now, the purpose of secreting acid in the first place, even at a very low level, is to maintain sterility of the GI tract, a sort of a protection against infection. Of course, when food enters the stomach, the food itself is not sterile, and therefore we would expect bacteria, etc., to be in the body, which of course a more aggressive secretion of acid would be warranted. The acid also helps break down a precursor enzyme, pepsinogen, into pepsin to initiate serious protein digestion. Now, coming back to the stimulation by way of these phases, when we say cephalic phase, what we mean is that the brain, sensing that food is in the oral cavity and is going to go into the stomach, the nasal appreciation, the oral appreciation of food would be manifest by way of the parasympathetics, i.e. the vagus nerve branches coming down to the stomach and stimulating the stomach lining by way of the acetylcholine secretion on the muscarinic receptors. This would start acid secretion and enhance acid secretion. We also have a gastric phase where these so-called G cells, which are located more towards the pyloric antrum, maybe even the duodenum, in the first part of the duodenum, these G cells secrete a hormone gastrin, which will stimulate parietal cell action. Now, the way it does it, there's a couple of different ways. It actually participates with the enterochromaffin cells to have sort of a combined effect, but we won't worry about that here. The gastrin actually induces a production and recruitment of the hydrogen potassium antiport channel to be introduced into the luminal membrane of the parietal cell, thereby enhancing more hydrogen ion secretion. Now, these enterochromaffin-like cells, or ECL cells, are reminiscent of neuroendocrine cells, let's say of the hypothalamus and the neurohypophysis, but these are located in some parts of the stomach mucosa as well as in the intestines. And what they do is, upon stimulation by some amino acids which have already formed, the cells would secrete histamine, and histamine in the blood would behave as a hormone, traveling to the stomach and stimulating the parietal cells into more secretion of acid by histaminic receptors. Now, just like you have stimulation of the acid, you also have inhibition of the acid, and the big inhibitors are the hormones secretin and CCK, which are produced by the duodenal lining. Once the stomach has the food going into the duodenum, amino acid and peptide releases from the ongoing digestion will already start to induce secretion of CCK and secretin, which will then serve to inhibit the acid release. The role of gastrin inhibiting peptide is kind of under scrutiny. It's thought now that the gastrin inhibiting peptide may actually not even be directly inhibiting anything. Actually, it's more of a function of inducing insulin to be produced. So we'll leave that off right now, but glucagon is definitely one of the inhibitors of the acid secretion. And curiously, there's one more, and that is somatostatin. Now, somatostatin is a inhibitor of growth hormone, and so it's something that you associate with the hypothalamus and hypophysis relationship. However, somatostatin is also produced in parts of the stomach lining, the small intestine. There's even pancreatic islets called, cells in the pancreatic islets called B cells. They also seem to produce this hormone called somatostatin, which has a inhibitory action on gastrin production, and therefore a inhibitory action on acid secretion. So in summary, we basically have acid secretion caused by production of hydrogen ion by the parietal cells. The way to stimulate acid secretion is by the so-called cephalic phase by way of ACH secretion from the vagus, the gastric phase by way of gastrin secretion from G cells, and the enteric phase, the secretion of histamine from the enterochromaffin cells. Okay? And of course, from the standpoint of inhibition, we have the following hormones. We have secretin and CCK, both coming from the duodenum, telling the stomach lining to slow down the production of acid. We have glucagon, which seems to also have an inhibitory action. It's coming from the pancreas. And then somatostatin, which is produced in multiple areas, stomach, intestine, and the D cells of the pancreas, also causing an inhibitory action when it comes to acid secretion. So this marks the conclusion of our brief discussion on acid secretion from the stomach. Thank you.